Hello friends, welcome to BMH Learning. This video is linked to the mechanism of apoptosis. A cell can be subjected to apoptosis due to several reasons, and depending upon various physiological factors, it may have three different types of pathways for controlled cell death. So apoptosis can be initiated from the signals of intrinsic pathway, extrinsic pathway, and perforin granzyme pathway. For better understanding, the extrinsic pathway has further two types, FAS and TNF pathway. Now all these mechanisms have got three stages. The last two stages are same for all mechanisms. But we are only interested in the first one, which is initiation. You will see every single pathway uses different ways to initiate apoptosis. In this lecture, we will only discuss how the apoptosis is initiated by intrinsic pathway. So in short, we say, in intrinsic mechanism, the mitochondria becomes leaky and starts losing its important proteins called cytochrome C. As soon as these proteins enter into the cytoplasm, they initiate the killing of the cell. So, what we have to learn here is how cytochrome C would come out of the mitochondria. Generally, the outer membrane of the mitochondria has proteins called BCL2 and BCLX. These are actually anti-apoptotic proteins. So that means their function is to preserve the integrity of the mitochondrial membrane by preventing apoptotic proteins from getting leaked into the cytoplasm. Now suppose cell is subjected to various stress forms causing mutations, which further lead to the DNA lesions or protein misfolding. Under these conditions, stress proteins are activated, which are collectively referred as BH3-only proteins. They are named as BIM, BIT, and BAT proteins. Now these BH3-only proteins serve two important functions. First is they block the function of anti-apoptotic proteins, that is BCL2 and BCLX. Second is to further enhance the apoptotic activity. BH3-only proteins stimulate two other pro-apoptotic execution proteins named BEX and BEC. These activated proteins create channels in the mitochondrial membrane. This action allows mitochondrial protein called cytochrome C to leak into the cytoplasm. As the cytochrome C enters the cytoplasm, it follows another protein called apoptosis activated factor 1 or APAF1. These proteins, when bind to each other, form a complex called apoptosome. The complex then recruits pro-caspase 9, which is then activated in the form of caspase 9. Then the activated caspase 9 binds to other nearby pro-caspase 9 molecules and activates them. Now, caspase 9 is an initiator caspase. To carry out apoptosis, these stimulated caspase 9 molecules bind to the executioner caspases called pro-caspase 3 and pro-caspase 6. This binding leads to the activation of the executioner caspases. Once the executioner caspases are activated, they cause apoptosis of the cell. That was all. Thanks for watching.